Hi, today's lecture is entitled Mutation. A second major force that produces evolutionary change is mutation. Mutation is defined as an actual alteration in the genetic material. If one allele changes into another, a mutation has occurred. Mutation is a molecular alteration, such as a change in the base sequence of the DNA. For a mutation to have evolutionary significance, the mutation must occur in the sex cells. This is because only genetic information that is in the sex cells is passed on from one generation to the next. Mutations that are in your autosomes, your non-sex cells, are not going to be passed on from one generation to another. The most important thing to remember about mutation is that it is the only way to produce new variation. It is the creative force of evolution, and as such, it represents the first stage of the evolutionary process. Whereas all the other evolutionary forces that we're talking about modify the genetic variation, only mutation produces new variation. So it is the most important, in my mind, of the evolutionary forces. One of the interesting things about mutations is that they're random. There's no way of predicting when a specific mutation will occur. All we can do is estimate the probability of a mutation occurring, but we cannot predict exactly when. The randomness of mutations means that mutations never seem to appear when they might be needed. For example, many mosquitoes have adapted to insecticides because a mutation was present in the population and it acted to confer some resistance for those mosquitoes that had that mutation to the insecticide. If the mutation had not been present in the population, the mosquitoes would have died out. The mosquito's need for a specific mutation had no effect on whether the mutation appeared or not. It was just by chance, by random luck, that this mutation actually existed. The conventional view of mutation has long been that mutations are harmful. But our view is that not all mutations are harmful. That some mutations are advantageous, some are neutral, and some are harmful. What I mean that some mutations are advantageous is that some mutations actually lead to changes that improve our survival and reproduction. For example, the mutation that conferred resistance to insecticide in mosquitoes led to changes that improved survival and reproduction for those mosquitoes. In recent decades, we have discovered that some mutations are what we call neutral. That is, that they do not seem to have any effect on survival or reproduction. And there's a tremendous debate regarding the frequency of these neutral mutations. Some mutations are in fact quite harmful, and they do negatively affect an individual's survival and reproduction. Mutations generally occur spontaneously in response to conditions within the body or in the environment. The exact cause of such a mutation is usually not determinable. That is, we usually don't know why it occurred. They may not have any effect on the phenotype of the organism if the mutant allele is recessive, so we don't actually always know when a mutation is present. There are a variety of ways in which mutations occur. Mutations can involve changes to a single DNA base, changes to a gene, or changes to an entire chromosome. Changes to a single DNA base are referred to as point mutations. They are the most common type of mutation and the most important source of variation in populations. A classic example of a point mutation is sickle cell anemia. Let me go back to the previous slide. Sickle cell anemia is a disease that is associated with a minute change in the hemoglobin chain. Okay, so we have all this chain of amino acids producing this protein hemoglobin, and it has a point mutation. Hemoglobin has 146 amino acids, and 145 are exactly like the normal ones in individuals who have sickle cell anemia. So they have one amino acid that is different in sickle cell anemia than they do in normal hemoglobin. This type of change, as I said, is referred to as a point mutation. Okay, mutations can also involve the addition or deletion of a base or larger sections of DNA. This is referred to as a frame shift mutation because all the other bases are shifted around the change. An example of a frame shift mutation, let's say we have a DNA segment GAA TGA. It produces these two amino acids, leucine and threonine. If we have a mutation where we add a base, we add the base C, cytosine, and we now have GCA, which produces arginine, and we have ATG, which produces tyrosine. This is a classic example of a frame shift mutation. Another type of mutation that occurs is when sections of DNA are added or lost during crossing over. The sections of DNA that are exchanged from the chromosome are not equal. Okay. Mutations also occur in terms of chromosome number. That normally in humans we have 46 chromosomes. But sometimes, due to a process called non-disjunction, the failure of the chromosomes to separate during meiosis occurs, and two members of a chromosome pair move together to the same pole. So non-disjunction is the failure of chromosomes to separate normally during meiosis. Here's one of the most common types of non-disjunction is Down syndrome, where we have 3 copies of the 21st chromosome. So here you see all your chromosomes, and they're all in pairs of 2, except number 21, where we have 3. You see it's a female because they have 2 X's. Interestingly, Down syndrome has a very, very high frequency. It's 1 in 1,000 live births. It is associated with advanced maternal age, where we have early initiation of meiosis in females and senescence of the gamete line, and 95% non-disjunction in ova, only 5% in sperm. What that means is that the majority of the situations are resulting from inaccuracies in meiosis in the eggs and not in the sperm. The risk for a woman aged 20 giving birth to an affected infant is only 0.05%, so less than 1%, but 3% of babies born to women 45 years or older exhibit trisomy. So most examples of Down syndrome are born to women over the age of 35. Okay. There are also non-disjunction problems that affect the sex chromosomes. These are extra or missing sex chromosomes. These syndromes generally produce individuals that are XXY, XO, so they only have 1 X chromosome, or they have 3 sex chromosomes, XXX. In all of these cases, the symptoms are less severe than those for abnormal numbers of autosomes. One of the most classic examples of chromosomal mutations involving sex chromosomes is Turner syndrome. As you can see in this first graph here, we have an X, but we have no Y. Okay, so individuals, instead of having the normal 46 chromosomes, have 45 chromosomes, and they generally develop as sterile females. In this example here, we have an X, another X, and then a Y. This is known as Klinefelter syndrome. So they are males, generally, with an extra X chromosome, and they generally have reduced fertility. Individuals with these types of symptoms, not symptoms, but syndromes, generally are referred to as intersex individuals because it's very hard to distinguish them as either male or female. I'm not saying they're androgynous, but in general, they have mixtures of characteristics of both males and females, making them more likely to, making it harder to say you're male or female. So just to reiterate, disjunction is the cause of improper number of chromosomes, and it results from failure in meiosis. The frequency of errors is actually quite high and has very serious consequences. It is known that one out of every 2 pregnancies terminates in a spontaneous abortion. This means that 70% of all spontaneous abortions are caused by an improper number of chromosomes. So this is a much higher rate of error than most people realize for meiosis. Genetic information can be altered at the chromosome level by 5 different processes. First is deletion, in which a segment of the chromosome is lost. Duplication, a segment is present more than once. Inversion, the segment is inverted. Insertion, a segment is added. And translocation, segment is moved from one part of the chromosome to another. All of these mutations occur through errors in replication during meiosis. A classic example of a chromosomal deletion condition is that of Cry-Duchat syndrome. It affects 1 in 50,000 live births. It's called the cat-cry syndrome, and it results from a short segment of chromosome number 5. It results in mental retardation, defects in facial development, abnormal development of the glottis and larynx are probably the most characteristic, and the end result of which is that individuals cry like cats. Another type of chromosomal deletion is Prader-Willi syndrome and Angelman syndrome. Prader-Willi is found in 1 in 25,000 live births. It's the loss of part of the paternal chromosome 15. Infants are very weak, but they begin to eat compulsively by 5 to 6 years of age, and they usually die of diabetes and heart problems at a very young age. Angelman syndrome is the deletion in the same section of chromosome number 15, but it's on the chromosome that you inherit from mother. It's also associated with severe mental retardation, no speech, happy disposition, excessive laughing, hyperactivity, a whole variety of syndromes. Duplication. Duplication is best. The best example of duplication is what are called trinucleotide repeats. Okay, and basically you have codons in your DNA that repeat for a period of time, and they're called trinucleotide repeats. So you have a sequence of 3 nucleotides. They repeat in tandem a variable number of times within a gene. Huntington's, normally you have certain sequences that repeat 6 to 37 times. For Huntington's, it's CAG, and you'll have CAG, CAG, CAG 6 to 37 times. But people that exhibit Huntington's disease, instead of having it 6 to 37 times, have it between 35 to 121 times. Individuals with myotonic dystrophy, these are commonly called tics, things like Tourette syndrome, they have the nucleotide repeat of CTG, and instead of having it a normal 5 to 37 times, they have it between 50 to 72,000 times. Imagine repeating the CTG 72,000 times within your DNA, and these are a very, very common type of mutation. We're finding more and more medical conditions are the result of trinucleotide repeats. The rate of mutation. The rate of mutation is extremely difficult to determine. There are several different base sequences that specify the same amino acid. So mutations that do not lead to any biochemical change, that are neutral and go unnoticed, make it really, really difficult to determine the rate of mutation. Mutation is also much more apparent with a dominant allele. If a mutant allele is recessive, you're going to need 2 copies of it in order for it to be expressed. This is very, very uncommon to have a mutation expressed twice. So you need a relatively high rate for that mutation in order for it to be expressed in the homozygous recessive condition. In addition, I want to reiterate, many harmful mutations result in spontaneous abortion before pregnancy is detected. So, as I said, mutations are very, very rare. The probability of a mutation at a given locus for a given sex cell is 0.001 to 0.00001. So here you see the rate of mutations. It's very low. Human chromosomes also, they have a ton of different loci. The exact number of loci is not known, but it's been estimated to be something like 100,000 loci on your chromosome. And the probability that a specific locus is going to have a mutation is low, but the probability that at least one locus will have a mutation is much, much higher. Assuming you have a mutation rate of 1 in 100,000 for 100,000 loci, that gives you a statistical rate of 63% that any chromosome will have at least one mutation. So if you have 1 in 100,000 mutation rates on 100,000 loci, it gives you a 63% chance of a mutation. So basically, my point is, mutation is very common within a single species and within a generation. We generally measure the frequency of mutations by what's called the mutation rate. The mutation rate is the proportion of gametes carrying newly arisen mutations at a particular locus. It is abbreviated mu, like a cursive U, okay? And we calculate the mutation rate for dominant point mutations. So you can calculate the mutation rate for dominant point mutations, but you cannot calculate the mutation rate for recessive ones because they're not being expressed. So examples of dominant point mutations is achondroplastic dwarfism, about 8 in 100,000. Neurofibromatosis, 1 in 3,000, so very high rate. Huntington's, 1 in 1 million. So this was the achondroplastic dwarfism. There was a study in Denmark, found 8 cases of dwarfs born to normal parents out of 100,000 births. So you have 8 dwarfs from normal parents out of 100,000 births. So the mutation rate is therefore 8 divided by 100,000. Neurofibromatosis. It's an autosomal dominant disease characterized by pigmentation spots and tumors of the skin and the central nervous system. Once again, 1 in 3,000 births are affected individuals, and they generally occur in families with no history of the condition, indicating the condition has a very high mutation rate. And Huntington's disease, once again, it's a dominant condition causing progressive degeneration of the central nervous system, and the mutation rate has been considered 1 in 1 million. So relatively high rate for Huntington's disease.